Right now, hiding inside the DNA of millions of people are the biological instructions for how to survive an ice age.
Which is, frankly, incredible to think about.
It's wild, right? We are talking about literal genetic code written by a human species that went entirely extinct over 40,000 years ago.
Yeah.
But today, those same ancient survival instructions are actually causing type 2 diabetes, and they are actively fighting against our most advanced modern medicines.
They really are.
So welcome to this custom deep dive. We are exploring a genuinely fascinating May 2026 Science Daily article. It details a massive genetic study by Riken, which is a major national research institute in Japan.
Right.
And we are going to look at how this ridiculously high-definition genetic map is uncovered like ghost DNA from extinct human species. It's completely shattered decades-old assumptions about human origins. And honestly, it's laying the groundwork for the entire future of personalized medicine.
Yeah, and it's a profound demonstration of biological continuity. I mean, we tend to view history as just this academic subject, like something confined to dusty archives or archaeological sites.
Right, something in the past.
Exactly. But what this data shows is that our deepest ancestral past isn't behind us at all. It's a physical architecture that is currently operating inside your cells, actively dictating your modern-day health and disease risk.
Which is just mind-blowing.
Yeah.
But, okay, to understand how this new genetic map upends everything, we kind of have to start with the old consensus.
We do.
For decades, the accepted history of Japanese origins was the dual origins theory. So the story went that the population basically stemmed from two distinct groups. First, you had the indigenous Jomon hunter-gatherers who had inhabited the archipelago for thousands of years.
Right.
And then much later, this massive wave of migrants arrived from continental East Asia. And they brought, like, rice farming and metalworking. So the two groups met, they mixed, and bam, that formed the modern population.
Yeah, and that theory survived for so long because it perfectly mirrored the visible archaeological record. I mean, when researchers looked at ancient settlements, they saw this sudden, very distinct shift in pottery styles and agricultural tools.
It's what was physically there in the dirt.
Exactly. And it is very easy to look at that physical evidence and just deduce a simple narrative, like, oh, an influx of new people bringing new technology. But that simplicity was really just an artifact of the limits of our tools at the time. Physical artifacts only tell you what people left behind, not necessarily who they were biologically.
Right. And that limitation is exactly what this Riken study, led by Chikashi Terao, completely broke through.
Right.
They didn't just analyze a handful of genetic markers. They performed whole genome sequencing on over 3,200 individuals stretching across all of Japan, from Hokkaido in the north all the way down to Okinawa.
Yeah, the scale and the method here are really what make the findings so disruptive, because historically, large-scale genetic studies relied on something called DNA microarray technology.
Okay, right.
And look, microarrays are highly efficient, but they only scan for very specific, predetermined snippets of DNA. You are essentially just checking a box, like, does this person have this specific, known genetic variant, yes or no?
Gotcha.
But whole genome sequencing, however, reads almost all 3 billion DNA-based pairs in a person's genome. So according to the Riken researchers, this approach yields roughly 3,000 times more information than those older microarray techniques.
3,000 times. Okay, let's unpack this for a second, because upgrading to 3,000 times more data requires a pretty serious mental shift.
It really does.
So think of the old microarray method like using the find function in a massive digital document just to search for 5 specific words. If those words are there, you highlight them and you just assume you understand the theme of the whole book.
Right, you get the gist, supposedly.
Yeah. Exactly. But whole genome sequencing is reading the entire document cover to cover. You aren't just finding the expected words. You are reading the typos. You're reading the weird margin notes. And you're finding whole chapters you didn't even know existed. You build a completely new understanding of the text.
I love that analogy. And that structural shift in how we read DNA is the exact foundation of their newly created database. It's called the Japanese Encyclopedia of Whole Genome Exome Sequencing Library, or JEWEL for short.
JEWEL.
Right. And just to clarify that acronym, the exome part refers to the specific 1 to 2% of the genome that actually codes for proteins, while the whole genome includes all the non-coding regions that regulate how those proteins are used.
Oh, okay.
So by analyzing all of it, and particularly focusing on incredibly rare genetic variants, Terao's team found the undeniable biological signature of a third ancestral group.
Right, the tripartite origins theory.
Mhm.
They actually found a third ancestor that appears to be linked to the ancient Emishi people of northeastern Japan.
Yeah.
But hold on, though, because if a completely distinct ancestral group was living there and contributing heavily to the gene pool, how did traditional historians miss them entirely? I mean,
Right.
weren't there historical records of these people?
Well, there were records, but, historical records are notoriously written by the victors.
Ah, true.
Yeah. The Emishi were this distinct group in the Northeast who actively resisted the centralized rule of the Yamato state for centuries. Over time, they were politically marginalized, they were assimilated, or just pushed further and further north.
Wow.
So when history is written by the central authority, groups like the Emishi get relegated to the footnotes. But this is the beauty of tracing rare genetic variants.
Yes, so.
While common genetic variants get mixed and widely distributed across a population over millennia, they sort of wash out. Rare variants, however, are highly localized. They haven't been smoothed out by time yet. They act as anchor points. So by tracing these rare, highly localized DNA changes, researchers could track fine-scale migration patterns that traditional history just glossed over.
That makes total sense. And when you look at those regional patterns in the Joule database, the level of diversity is honestly staggering. Like, the original Jomon hunter-gatherer ancestry is highly concentrated in the south, specifically in Okinawa, making up about 20.5% of the genetic signature there.
Right.
But then when you move to western Japan, that Jomon ancestry drops sharply to just 13.4%.
Which is fascinating because it perfectly maps onto a completely different set of historical migrations. The data reveals that people in Western Japan share much stronger genetic ties with Han Chinese populations.
Okay, so a direct link to the mainland.
Exactly. And this isn't just some random distribution. It aligns perfectly with major migration waves from continental East Asia that occurred much later, roughly between 250 and 794 CE.
Oh, the Kofun period. Those migrations were essentially importing the foundation of modern civilization into the region.
They really were.
You're talking about the introduction of Chinese-style centralized government systems, the integration of written language, formal education. So the people flooding into Western Japan were rewriting the cultural and political rules while simultaneously rewriting the region's genetic code.
Yeah, and the Joule database allows us to see that in high definition. You have the Jomon ancestry anchored in the south, the Emishi-related ancestry concentrated in the northeast, and the Han-affiliated ancestry dominating the west. The modern genome is literally a layered map of ancient borders and centuries of human migration.
It's like an archaeological dig inside our own cells. But if this incredibly high-resolution DNA map can spot an isolated, marginalized tribe from northern Japan from a few thousand years ago, what happens when you stretch the timeline further?
It gets wild.
It gets so wild. What happens when you use that same technology to look back 40,000 years? You don't just find missing ancestors, you find entirely different human species. The Riken study identified 44 archaic DNA regions in modern Japanese populations that were inherited from Neanderthals and denizens.
Right. And just to set the context for anyone who might need a refresher, Neanderthals and Denisovans are our extinct evolutionary cousins.
Right.
So tens of thousands of years ago, when early Homo sapiens migrated out of Africa and into Eurasia, they didn't just share territory with these other human groups, they actually interbred with them.
Ah.
And as a result, segments of Neanderthal and Denisovan DNA were folded into the Homo sapiens genome, and they have been carried forward through the generations ever since.
And the study notes that many of those 44 ancient DNA regions are entirely unique to East Asians. But here's where it gets really interesting. Because we're about to discuss how these ancient genes are linked to severe, life-threatening modern diseases. So my question is, if natural selection is supposed to be this ruthless editor that removes harmful traits, why didn't evolution just weed these archaic sequences out?
That's a great question.
Right. I mean, if a Denisovan gene is actively harming people, shouldn't it have vanished from the gene pool over the last 40,000 years?
Well, if we connect this to the bigger picture, we run into one of the most counterintuitive mechanisms in evolutionary biology. Because evolution is entirely contextual.
What do you mean by that?
A biological trait is only harmful or helpful based on the specific environment in which it operates. Evolution doesn't optimize for your long-term health or your comfort in a modern city. It basically just selects for whatever keeps you alive long enough to reproduce in your immediate wild environment.
Ah, okay.
So a genetic variant that provided a crucial survival mechanism during a brutal ice age can easily become a massive liability in an era of grocery stores and central heating.
Wow. It's almost unsettling to think about. We're carrying around biological hardware engineered for a world that literally doesn't exist anymore.
Exactly. And the source material provides a classic example of how this plays out in real life. Previous research has shown that modern Tibetans carry a specific Denisovan-derived version of a gene called EPAS1. And that ancient gene significantly alters how the body processes oxygen. Now, in a low-altitude environment, that might not matter at all. But high up in the Himalayas, where oxygen is scarce, that Denisovan gene is literally the difference between thriving and severe hypoxia.
So it was a massive evolutionary advantage.
It was profound. But on the flip side, we also know that certain Neanderthal-derived DNA segments heavily predispose individuals to severe respiratory complications from COVID-19.
Wait, really?
Yeah, the immune response that kept a Neanderthal alive against prehistoric pathogens just severely overreacts to a modern virus.
That is insane, which actually brings us right back to the new data out of Japan, because this study adds a massive piece to that puzzle. Tarao's team found a specific Denisovan-derived region inside a gene called NKX61.
Yes, the NKX61 gene.
And this piece of archaic ghost DNA is strongly associated with type 2 diabetes.
And the implications of that finding go way beyond just predicting who might get sick. The Riken researchers observed that this specific Denisovan genetic fragment may actually alter how a patient responds to the modern drug semaglutide.
Semaglutide, wait. The major diabetes and weight loss medication?
That's the one.
How does a 40,000-year-old gene interfere with a drug engineered in a modern laboratory? I mean, how is that even possible?
It all comes down to the physical shape of cellular receptors. See, genes are essentially blueprints that tell your body exactly how to build proteins and receptors.
Okay, cracking.
Semaglutide works by mimicking a hormone that targets very specific receptors to stimulate insulin production. But if your Denisovan DNA slightly altered the blueprint for those cellular pathways, say perhaps to help your ancient ancestors store energy and survive lean, freezing winters, well, the architecture of your cells is fundamentally different.
Oh, I see where this is going.
Right. So a modern pharmaceutical designed for the standard quote-unquote human receptor simply might not bind correctly. It might not trigger the expected response at all.
So a pharmaceutical company can spend literally billions of dollars perfectly designing a key, only to find out that a caveman changed the locks 40 millennia ago.
That is exactly what happened, yes.
That is staggering.
Yeah.
And it wasn't just Denisovan DNA, right? The team also identified 11 distinct Neanderthal-derived genetic segments connected to a whole spectrum of modern conditions, like coronary artery disease, prostate cancer, rheumatoid arthritis.
Yeah, and identifying these connections is really the critical pivot point of this entire field of research. Cataloging archaic DNA isn't just some academic exercise in mapping human history. It is a biological necessity for modern medicine.
Absolutely.
When we understand exactly how these ancient sequences operate at the molecular level, we can finally stop using generalized treatments. We can begin developing therapies that actually account for an individual's unique biological architecture.
And if you're listening to this and wondering, like, okay, this is fascinating ancient history, but how does this actually affect my next doctor's appointment? This is exactly where the Riken study shifts from history to immediate practical medicine.
Right.
The Joule database is actively identifying dangerous mutations right now. The team pinpointed potentially harmful variants in a gene called PTPRD. And if you carry variants in this gene, you face significantly higher risks of hypertension, kidney failure, and myocardial infarction.
They also isolated specific loss-of-function variants in two other genes, GJB2 and ABCC2. And these are strongly linked to hearing loss and chronic liver disease.
Loss of function basically meaning the gene doesn't work, right?
Exactly. A loss-of-function variant means the genetic construction is essentially broken or switched off, which prevents the body from producing a necessary protein. So by identifying these specific broken pathways within the Japanese population, doctors can screen for these risks well before the diseases ever manifest clinically.
So what does this all mean for us? Why is it so crucial that they are isolating variants specific to Japanese populations? Think about it this way. Imagine you manage a vast, globally diverse fleet of cars.
Okay, I like this.
You have vehicles built for Arctic tundras, desert dunes, dense cities, but the only tool you have in your entire garage is a single repair manual written exclusively for one specific European model.
Yeah, that's not going to work.
Right. If a completely different vehicle rolls into the shop and you try to fix it using that one manual, not only will the instructions be completely useless, but you might actually destroy the engine trying to apply them.
That is spot on.
Trying to practice global medicine today, using genomic databases that only understand one type of ancestry is exactly like trying to fix that diverse fleet with one highly localized manual.
This raises a really important question regarding the structural foundations of modern science. The analogy of the single repair manual is entirely accurate, because for decades, the vast majority of large-scale genomic databases have heavily, almost exclusively, focused on individuals of European ancestry.
Which is a huge blind spot.
It's a severe blind spot. Because researchers have primarily studied European genomes, their understanding of disease risk, genetic predispositions, and drug efficacy in non-European populations has been profoundly limited.
Because we've literally only been reading one version of the manual. We haven't had the data to see how the machinery works in anyone else.
Precisely. And this is exactly why Chikashi Terao's work with the Joule database is such a watershed moment. Expanding this genomic library with comprehensive Asian genomic data is a vital, non-negotiable step toward equitable, personalized medicine.
It has to happen.
It does. As the researchers themselves emphasize, building out this data is essential so that the benefits of precision medicine can eventually reach everyone, regardless of their ancestral background. I mean, knowledge has limited utility if it only applies to a fraction of the population.
Right.
By understanding these unique population-specific genetic traits, the medical field can finally abandon that dangerous one-size-fits-all approach and begin treating the actual biological reality of the individual patient in front of them.
When you step back and look at the sheer scope of what we've covered today, it is really remarkable. We started with a shift in technology moving from scanning for a few genetic keywords to reading the entire 3 billion-letter manual of human DNA.
Yeah.
And that technology completely rewrote the history of Japan, proving the population was forged by 3 distinct ancestral lines, including the forgotten Emishi people.
It's amazing.
And then it looked tens of thousands of years deeper, uncovering the archaic ghost DNA of Denisovans and Neanderthals that is secretly dictating our disease risks today and fighting our modern medicines. And ultimately, it exposed the desperate need to build inclusive medical databases so we can actually treat the entire human race.
It really forces us to recognize how deeply tethered we are to our evolutionary past, which I think leaves us with something to consider long after this discussion ends. Your genome isn't just a biological blueprint. It is a living archaeological dig. It holds the ghosts of extinct human species, the borders of ancient empires, and the evolutionary trade-offs of ice ages. So if a new technology tomorrow could read your DNA at an even deeper level, what invisible history is currently writing the script for your health, just waiting to be discovered?
Wow. That is definitely a thought to carry with you. Thank you so much for joining us on this deep dive. Keep questioning the world around you, and we'll see you next time.